This is episode 66 of the Immunology Podcast, Immune Cell Interactions with Dr. Sophia Liu. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology Podcast. We have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today we have Dr. Sophia Liu from the Ragon Institute on the podcast to talk about her research on immune cell interactions in tissues. We've also got our usual roundup of recent highlights in immunology news coming up. But first,
it's almost time. Almost there. IUIS 2023. We'll see you in South Africa at the International Congress of Immunological Societies, happening in less than a month. Jason's so excited. Dear listeners, you can check out the program and choose which sessions to attend at IUIS2023.org.
I am really looking forward to the visit. I am not looking forward to that flight.
It's the first time that you cross, two hemispheres, to the east, to the west, to the south, to the north.
At once, yes. I have not done a twofer in one trip before. I've done up and down and left and right. I just haven't done diagonal before.
Don't worry, you'll survive. You can trust me. I've done it many times.
Yeah, that's because I'm getting a bougie little place to sleep, so I don't have arthritis when I get there.
Yeah, we already know you're getting business or whatever. We don't want to know. Most of our listeners will be flying economy, so get a grip, Jason. Just be nice.
I have to be able to walk when I get there. I'm old.
Oh yeah, yes.
We need the STEMCELL Podcast to tell me what to inject in my hip so that I can regenerate all my cartilage that's clearly gone.
Oh, wouldn't that be nice? Wouldn't that be nice? Yeah. So very excited. So where should IUIS be? I think it's every 2 years, the conference, or I don't know how often it is.
I either want New Zealand, Japan, or Italy. Those are my three places I want to visit next.
Just exactly the same places. So yeah, Japan or Italy. Okay, for different reasons, I guess.
Well, no, the same reason. Food and culture.
I would love New Zealand.
Food and different culture, but I want to go for the food and culture.
I mean, you do know that New Zealand are called Kiwis, but there's a lot of Kiwis there.
But I want to go to New Zealand to see the Lord of the Rings stuff.
Yeah, of course you do. Actually, we had a marathon, Lord of the Rings extended versions marathon at home.
When did you do that to yourself, and why?
Well, to be honest, my boyfriend did. I just watched the first, and then I had stuff to do because I have things to achieve.
Is that like over 24 hours?
No, it was like 12 hours. It was not that bad. They left right after midnight.
You know what's longer than 12 hours?
The flight from the US to South Africa. Oh my God. You know what? Dear listeners, please do comment on our Twitter feed how long your flight to IUIS 2023 will take, so that Jason stops whining about having to take a long flight for once in his life. You are just too used to going, just the transatlantic US route. You forget that the world is big.
I fly to Hawaii not infrequently, and from Boston, or from the East Coast to Hawaii is East Coast to LA, then LA to Hawaii. That's about 13 hours a trip. So I've done it with small children. It's just not one flight. Then
he was like 40 hours. He was 14, but he felt like 40. Oh,
there was a 5-hour layover at LAX.
Oh God. Actually, I don't know. Is it? I have to say that usually American airports, I don't like them too much. I had a very disappointing experience in places like LaGuardia. Are horrible. I don't know how LAX it compares to it.
I love the Yotel in Amsterdam. We went to France. We flew Philly to Amsterdam and then Amsterdam to Bordeaux instead of driving. And there was this Yotel, and so we just rented one of those by the hour hotel rooms where like the toilet and shower were like one unit type of, like, if your feet went into the shower, the toilet, and like the bed could flip up and the chair could come out. It's kind of cool and robotic, but like my wife and I just passed out in the hotel for a couple hours.
I mean, that's the whole point. And this one level of, on top of like capsule hotels. So I think it's good.
Oh yeah. The hotel was great. All right. Am I going first again? Usual. We still have an obligatory, but not COVID, coronavirus, non-COVID.
Oh, is there such a thing?
There are.
Please tell me more.
Did you know the common cold, or some of the common colds, are caused by coronaviruses?
Oh no. Oh
But
no, my life is a lie.
But wait, there's more. We don't know a lot about all of them, but we've decided it may be good to learn more. So in today's news, in a Nature paper accepted October 18th, 2023, it's not actually released yet. It is TMPRSS2 is a functional receptor for human coronavirus HKU1. First author is Neil Saunders. Last author is Olivier Schwartz. So high level here, there's a bunch of viruses that cause and excuse me, the other last author is Julian Buchreiser. So the caveat of this is all that lots of viruses cause the common cold. Of the coronaviruses, there's HKU1, 229E, NL63, and OC43. And kind of in a post-COVID world, we're like, oh my God, we should probably learn more about coronaviruses. And we kind of figured out how to monitor all types of viruses much more rapidly and build some infrastructure. People are starting to look at other things that make people sick, like the common cold. And one of these is this HKU1. And we know that angiotensin converting enzymes that's what COVID uses, is used by 229E, and other ones use combined cells through acetylated salicylic acid, but the protein receptors for endocytosis aren't known. And so they found in this paper that transmembrane serine protease 2, TMPRSS2, is the receptor that does it. And now I'm done. So that's what they found. That really is the punchline. The paper really is a study on how they showed that using combinations of knockouts, pseudoviruses that had it or didn't, cells that had the receptor and didn't. They made nanobodies. If you don't know what nanobodies are, they use llama nanobodies. That's just the heavy chain, and llamas make those. So they tried to find different regions of the mutant. They did find that the catalytic activity of the protease was not required, though. So the enzyme binding to the spike generates it. Other proteases didn't, but inactive protease still allows for infection. I think that's the important part, is this isn't a catalyst, enzyme climatic cleavage. It's the spike binds to it, and that induces endocytosis. And so antibodies block it. So you could have an antibody to the common cold in sick people, but fundamentally knowing the receptor that leads to infection by an agent of the common cold and starting to figure that out is a pretty good advance in, virology.
Very interesting. It's fun to see how fun is not the word, but yeah, how there's so many receptors, so many molecules on the surface of cells that these viruses can hijack to get into the cells. I wonder sometimes if these molecules, why are they chosen? Like, is it because of their distribution in the respiratory epithelium, therefore would make a better type of cell to be infected, because then you can cough the virus to the next person? What is behind the which receptors are chosen by this virus?
I think that's a really good question. I don't know. I don't think we know yet. But epithelial origin tends to be a common theme for these.
Mm-hmm. I want to move on to the next paper because I don't think you're gonna like it, and that's why I chose it. Because one of the conclusions, I'm pretty sure you're gonna have an issue with.
All right, lay it on.
So prepare. So this paper came out in PNAS, and it's called "The Total Mass, Number, and Distribution of Immune Cells in the Human Body." First author Ron Sender, and the corresponding author is Ron Milo from the Weizmann Institute in Israel. And what they did basically is they did this huge kind of meta study looking into a lot of kind of previously already existing data to come up with a quantification of kind of rough subsets of immune cells in different organs and different lymphoid and non-lymphoid organs and the blood, to kind of characterize and give some kind of numbers to the immune system. And I think it was very cool because, sometimes I don't think we have a good grasp on like the percentage of us that is different parts of us. So basically what they did is, as I mentioned, they really dig into the literature. They looked into previous reports. They looked into histology, performing various types of publications. They looked into imaging data, and they kind of tried to estimate things such as cell density, cell amounts, and for different types of tissues. They also used something that I thought was very interesting, which is a deconvolution based. So a methylation atlas based on the convolution of full tissue methylomes, so to say. So because there are different cells that do have some kind of characteristic methylation, and there is kind of several methods that allow to kind of and extrapolate how many cells of different types were in a mix of methylated DNA. And so they also use this. So based on the methylation patterns of a sample tissue, they could identify subcellular subpopulations, in particular immune cells. And so with this information, they did a lot of kind of statistics and analysis. So I think that's kind of all the boring stuff. But they did a lot of kind of try to put some order into all this data. And they came to a couple of numbers that I'm going to share with you. So I'm going to quiz you a little bit. How many cells do you think they found? How many do they estimate that a reference 73 kilogram man that is like 150 pounds for you has?
Because that's a normal sized man in the Western world. Totally normal.
I think, I don't know, how much do you weigh? No,
more
more,
than
less?
73 kilos.
More than 73 kilos. Well, maybe the American average is different. I don't know. Well, that was mean. Sorry.
So
give me a number.
Total immune cells in the body.
Yeah.
That's what we're going for. Total immune cells.
Yes.
Well, I think we're like 1 trillion cells or something. I think is a total number for a human. So I'm gonna.
So 1 trillion.
No. So humans are 1 trillion cells. So I'm gonna.
So that is 10. The trillion would be 10 to the.
So humans are 1 trillion. So that's 1 times 10 to the 12.
10 to the 12, because trillion does not mean, like, the word trillion means different things in different languages sometimes.
Okay. So 1 times 10 to the 12th is what humans are supposed to be.
Yes.
Roughly. And so I'm going to say immune cells are roughly 10% of the entire body in terms of number. Actually, probably more than that, because they're tiny compared to like a muscle cell, which is gigantic. So I'm going to go 3 times 10 to the 11th. We're 30% by number immune cells, even though by mass, like our muscle cells way outweigh immune cells. But that a muscle cell like goes from your knee to your hip. One cell does. So the size is different. So 3 times 10 to the 11th.
Yeah. So according to their calculations, you are missing on one order of magnitude. So I don't know if they do an estimation of a total cell numbers in the body, but I don't think they do. But they estimate 1.8 times to 12 immune cells in the reference person's body.
So I just went into the Googles, and the new estimate from old stuff is there's 36 trillion cells in a human male.
Okay, so a little bit less than 10%.
So my math of about 10%, but that I went up, but that was closer. I was just off on the total amount.
Yeah. And this is where you're not going to like it. Where do you think most of the immune cells reside?
Probably the intestine, or probably the mucosal areas totally.
Are you sure? Is that your final answer?
Well, no, it's probably actually the skin.
No. I mean, the answer actually is pretty obvious.
Marrow.
Yeah. So They
Does that
do.
count progenitors and differentiated? Is that all in, or is that only differentiated?
I don't know how they define the cells in the bone marrow. So they suggest that the bone marrow contains 7.4 times 10 to the 11 cells. And that comprises 40% of all immune cells.
I mean, if you count all of the progenitors in that, I totally believe it.
Mm. And then they say that another 39% is actually in the lymphatic system, unsurprisingly. And that skin, lungs, and GI tract is only about 3 to 4% of all the cells in the human body, and less than 2% are in the blood. And I think the point that they make is that it's in cell numbers, but oftentimes many of the cells that are maybe in terms of mass, it might be bigger because oftentimes you have cells like macrophages, like which are very big, or plasma cells which are a little bit bigger, and then they might actually represent more a little bit more mass. So yeah, what else? So what do they say? So as I mentioned, most of the cells in the bone marrow and the lymphatic system. And actually, interestingly, about 70% of cells in the bone marrow are neutrophils. Oh, there's a lot of neutrophils in
Yeah,
there.
that's not surprising.
Well, it's not surprising, I know, but it's I always ignore the neutrophils, and I think this shows me that I'm making a mistake. And then I think it's also interesting. Of course, they also bring forward the fact that there's a lot of differences in the distribution of different immune cells. And for example, while macrophages might be a minor fraction of the immune cells in tissues such as bone marrow, they are a huge contributor to other tissues, to this immune cells in other tissues, particularly in the liver. So in the liver, macrophages comprise 70% of the immune cell populations. And in the lung, that number apparently is around 40%. So interesting, because also macrophages are a lot larger than leukocytes or lymphocytes and things like that. So they actually, when it comes to mass, they might represent a larger mass in these organs. They, for example, when it comes to the GI, the gastrointestinal tract, 70% of plasma cells in the body are there. Also, not surprising given that we have so much, immunoglobulins in the gut, and there's such a strong humoral response over there, so makes sense. And also, when it comes to the other cells, for example, mast cells, NK cells, they are kind of distributed. They're not in any particular organ more than other cells, like, for example, neutrophils, eosinophils, and monocytes, basophils, which are mainly found in the bone marrow, are not so much circulating in other places. And what I also thought was cool, they estimate that there is a total of 1.2 kilograms of immune cells in the standard adult body, and although the bone marrow and the lymphatic system have 40% of the cell numbers, they have less than 30% of the mass each, because of they're usually smaller cells. And then when it comes to the immune system in the gut and in the skin and other tissues, they are about 23% of the total immune cell mass, not necessarily the immune cell numbers. And what I also thought was cool, what do you want to say?
I was gonna say that makes sense, right? Like the math and the numbers don't line up necessarily.
Yeah, exactly, right? Because, and usually it is the fact that certain cells like macrophages are huge compared to other cells like lymphocytes, which are not that big. And so then they kind of extrapolate this to the average female person, and they say about 1.5 times 10 to the 12 immune cells in the reference female, with about a kilogram for a woman of 60 kilos, and that a child has about 1 times 10 to the 12 immune cells that weigh around 600 grams. I think also they have less of these more heavy cells at the early age. So now I hope you hold these numbers in your head for next cocktail party. You can always bring the fun facts.
Fair. I will. Now, neutrophils, when people are sick,
Mm-hmm.
the neutrophils mobilizing from their marrow into their blood is one of the main signs of it early on.
Yeah, because you usually don't find that many in the blood.
Right.
Mm-hmm.
But then they spike. You're like, whoa, where'd those come from? The marrow.
They were sitting there all along.
They were there the whole time.
Yeah. So well, I thought it's a very cool. I mean, um, it's a fun paper to watch. To kind of bring forward like these numbers that you're like, huh, 1.2 kilos. It's kind of cool. It's kind of cool.
And a below average sized male.
In a below average size male, exactly.
All right. Well, to keep on the theme of immunological markers or understanding some numbers behind things versus, say, mechanism. This is immunology responses to gut bacteria associated with time to diagnosis and clinical response to T cell directed therapy for type 1 diabetes prevention. First author is Quinn-Hugh Ji. Last author is Jane Danska. It's in Science Translational Medicine. And it came out the 25th of October. All right, so this is what's cool here, folks. There is a trial of there is this study called, let me get the acronym. It has a, like all clinical trials, has a very fun acronym, the TrialNet 10 or T10 study. Small study, but they used a anti-CD3 antibody called tepalizumab in patients with early not early onset, but early signs of type 1 diabetes, to ablate their T cells. And this trial showed that you can extend the time to diabetes out by several years. Diagnosis of 32.5 months. So almost 3 years delayed diagnosis with this drug. But then they looked at some other stuff from this trial. And that's what's really cool. And so here it is, Brenda, all at once, and then I'll wait for your reaction. There is something called, anti-commensal antibodies in your body. IgA, IgG1, IgG2. IgG2 anti-commensal antibodies to specific bacteria are predictive of how well you will respond to therapy. And if you didn't get therapy, also your prognosis. So this is the antibodies you had before you started on therapy that remain the same throughout therapy, do not change after therapy, and they do not affect any of the antibodies found during the disease state, right? So, there's all these antibodies that are associated with diabetes. None of that changes. The only thing that matters is, do you have these antibodies at a high level or low level? And if you have them at a high level to specific microbiota, which I will give the names for in just a second, if you have them to specific microbiota, you are more likely to be a responder to the therapy. So one of these is Bifidobacterium longum. Another one is E. efficalis. And another one that is interesting, D. invisius. D. invisius only matters depending on your HLA type. So the effect on that one is restricted to HLA type that you have at baseline. And these predict your response to a therapy and your general trajectory of disease to begin with. And there you go. And then they did another test case with other people who didn't get therapy and looked at their predicted responses, and they saw the same story essentially. So essentially, it predicted the entire response, which is crazy.
So are you telling me the bacteria are giving me diabetes? Is that it?
No, I'm telling you your antibodies to the bacteria, because your immune system is naughty, is predictive of you having worse outcomes. But similarly, anti-CD3 therapy is better for you in this case.
Because then I guess there's more of T cells that are being targeted by the CD3. There you have a kind of a...
No, your total immune cells aren't different. It's just, if you have a lot of these antibodies, you're more likely to have worse disease, faster onset diabetes, type 1 diabetes. But the therapy, the anti-CD3 therapy, works better in you than people with slower onset who have lower levels of these antibodies.
Right. But the T cells are in there. I mean, those T cells somebody has to be helping those B cells make those antibodies.
T cells are absolutely naughty little suckers involved in the disease. because the anti-CD3 antibody prevents the diabetes, but in people with, particularly people with a lot of antimicrobial antibodies.
Wow. It's just
It's not necessarily
how many
causative. It could be a
levels?
biomarker, right? It could be a
Yeah,
biomarker that if you're making anti-commensals, you're probably also more likely to hate your own pancreas, maybe.
that's also a way. Okay. Yeah.
And so it could be correlative. It's not necessarily causative. It's just that if you have this as a marker, you're more amiable to therapy. And biomarkers really matter in clinical therapy these days.
Yeah.
So it's quite the good biomarker.
All right. Well, it's very interesting. Very cool that they can really delay the onset of diabetes with this treatment.
3 years is quite a long time.
All right. All right.
Like, 10% of some of our lives, almost ish.
10%. Yeah. Yeah. Not for long. We have a paper about T-Rex. Like, I think it's a good way of ending this segment. Jason is shaking his head. Um, so in this publication, it's called "Resident Regulatory T cells reflect the immune history of individual lymph nodes." First author Ann Kaminsky from corresponding authors Oliver Pabst and from the University of Aachen and Milas Uger from the University of Würzburg in Germany. And in this publication, they used an interesting mouse model. Sorry, published in Science Immunology. They use a mouse model to study resident T cells in the lymph node using a reporter mouse that has a reporter that expresses a histone H2B fused photoconvertible protein called Dendra2, which can be photoconverted using violet or ultraviolet light, which basically allows to kind of shine the light on the lymph node and mark all the Tregs in there. And then you can see whether those cells are staying there or not. And then you can define resident Tregs based on the fact that they're still there in the lymph node after 24 hours. They cannot do much longer because this frequency of the marker, they use this DREG protein. It's not super stable, I guess, but they are probably underestimating a little bit the amount of positive cells that are staying behind, but they can use this in a way to show that these cells stay there. They are in the lymph node, they're not going anywhere. I think we have this idea that often lymph node is kind of a passing, is a pit stop on your way from to whatever. And they argue in this paper that actually you have a population of the focus on the Tregs that actually are still there. And they show that even 8 weeks after photoconversion, the photoconverted lymph nodes still contain higher frequencies of these fluorescent positive Tregs compared to other unconverted lymph nodes and the spleen, showing that there's like these cells in a particular lymph node, there are certain cells that have a tendency to stay there. And so this is how kind of they define residency in a more of it's not so much on the markers that they're expressing, but on the fact that they're actually there after X amount of time. And they show that they're not short-lived cells. There are these effects, they're resident there and they stay. They don't necessarily express the what we consider traditional residency markers like CD69 and Ki67. But they do seem to represent a distinct population of memory cells in the lymph node. So they do a lot of assays looking into showing that the cells they're comparing them with the conventional cells in the lymph nodes, showing some things about the kind of the residence memory like phenotype that they have, whether they have it or not. And they show that CD69 and Ki67 is not sufficient to characterize this population. There's some heterogeneity within this population. They don't all express CD103 or CD101, which are also usually associated with tissue residency. And I guess, on the one hand, I'm not super surprised because, I mean, tissue lymph node residency, does it have to be the same as tissue residency, non-lymphoid tissue residency? Maybe it's not necessarily the case. But in general, they kind of characterize these cells, and they seem to find that um, they express lower levels of certain genes that are usually associated with egress from the lymph node, so KLF2 and S1P or 1. So this is probably why they are sticking in the lymph node and they're not leaving the lymph node, as you would expect from usually naive cells are very keen on circulating, but not this resident Tregs. They don't seem to, they also use some markers that are used to differentiate, thymus derived from peripherally induced Tregs. They use neuropilin, Helios, and ROR gamma T. And they don't find differences between the resident and circulating Tregs, effector Tregs, when it comes to Helios. So this Helios, neuropilin, or ROR expression. So it seems that either peripherally induced or thymus-derived Tregs can acquire this migratory property and this residency in the lymph node. They don't seem to need specific TCR signaling to stay in the lymph node. So I think that's also interesting to know, because Tregs usually do need. They also are kind of used to receiving TCR signaling, but it doesn't seem to be indispensable for their residency in this lymph node. And when it comes to a repertoire, they also seem to have a distinct TCR repertoire, which might, in a way, reflect which lymph node it is, whether it's a lymph node close to which tissue. And they do some experiments with mesenteric lymph nodes, and they show that there's differences in the repertoires between lymph nodes located in different areas of the mesenteric mesenteric lymph nodes. For those who don't remember, they look like little beads next to each other. They're long and they can be kind of draining from different parts of the intestine. So they do see differences in the TCR repertoires in the different parts of the mesenteric lymph nodes, and these resident cells, the cells residing in different lymph nodes. So I thought it was pretty interesting, because I don't think we usually think about residency in lymph nodes. I often think of lymph nodes as passing areas, but there seems to be this population that by de facto is staying there, because they stay in it and is still there several weeks after. So what exactly that means and whether that really translates to the human situation is unclear. These are mouse experiments, but yeah, Tregs, it would make sense that they're there, taking hold in specific lymph nodes to keep immune responses. After all, immune responses would be initiating lymph nodes. So if you have them already there, it makes sense that they stick there to prevent conventional cells from going crazy.
So this is really just describing the mechanism by which they stay in their cage.
Yeah.
Despite all my rage, I'm still a regulatory T cell in a cage.
No, they are the cage. They keep them. They're kept in the cage to make sure that nothing happens in the cage, I guess. Yeah.
To keep the rage in the cage, they stay in the cage. That's fair.
There you go.
They're like cops in the prison, in the cell with the people. Fair enough.
Sure. Let's go
We're
for
just going
that.
to drive this analogy into the ground because I won't let go. All right, no. Well, in a second, we're going to be speaking to Dr. Sophia Liu at the Ragon Institute here. But before we get to that, are you looking for in-depth information on cell separation? Download the Cell Separation ebook from STEMCELL Technologies. It is a practical guide on everything you need to know about cell isolation techniques. It includes a collection of protocols. Visit stemcell.com/cell-separation.
For our second part of the podcast, we are, of course, joined by a great researcher. Today, we have Dr. Sophia Liu. She is core faculty and early independence fellow at the Ragon Institute of Mass General, MIT, and Harvard. And I'm very excited to talk to her about some really interesting research she has done on understanding T cell and B cell development kind of in a spatial dimension. It's really cool research. And also I would like to also ask her a little bit about her career. She is, as you say, a very early independent, a very young PI. I think a very an inspiration. So it will be very interesting to hear about her research and her career so far. So, Dr. Liu, thank you so much for joining us today.
Hello, thank you so much for having me. I've listened to a few of the episodes on the podcast and over the last few years, and I never expected to be a part of it. So it's really, really exciting. And I was just thinking earlier that the whole idea of a podcast is quite thrilling. It's like the nakedness of doing a chalk talk, but worse in a way. um, so I'm really looking forward to talking about some immunology with you folks. And as for being young, I had a rotation student the other day comment about how I had a gray hair. And so young PIs can feel old too. Yes.
Oh, my hairline receded far before I was your age, so I empathize with this. That's what grad school does to you, is it ages.
Yeah, Gen Z, brutal,
Yeah. Just tell him, wait until you're done with your PhD and we'll talk.
Oh, yeah.
Okay, so why don't we get right down to it? So maybe we can talk a little bit about your research interest, and I think I was very impressed by your fairly recent paper that you published in Immunity, in which you're doing spatial mapping of T-cell receptors, and you're using this in a number of different contexts, both in cancer and in thymus development, and you're having I thought it was really cool because oftentimes we don't pay enough attention to the where the T cells are because it's hard. So why don't we start talking a little bit about that? Why do you think it's important to map T cells in space?
Yeah, so I will say I'm an engineer and biophysicist by training. So almost certainly you know more immunology than me. But as someone who's really focused on building tools, I was really interested in looking at T cells because they're, in a way, at the core of many of these tenets of immunology. And so, abstracting immunology, taking it one step back, basically all of the research questions that we have try to hit at the idea of how do we generate so much diversity? How do we generate immunological memory? How do we kill things? And how do we distinguish between self and non-self? And, at the heart of these questions is something T-cell related. And so I was really interested in building a tool that could let us see where these cells are in tissues, because fundamentally immune cells need to physically interact with target cells to elicit some change. And so that was the part that was really exciting for me. I was doing some single cell work before, but these questions of where are these cells, how are they affecting other cells, sort of led me down this path of building tools to look at cells in tissues.
So, being an earlier life a biophysicist by training at a different institution up the road from where you're at, out in Brandeis, I love when you can use fancy science magic to understand things. So I was wondering if you could tell us a little bit about your specific science magic. So what are the tools you're using that let you do the work you're doing? And we can dive a little deep here and talk fluorophores and probes and all
Ooh,
you're doing to get stuff going. So fire away.
yeah, let's do it. So I guess, again, I think people ask why, is RNA so popular of a space, right? And it's because it's so easy to sequence and read out RNA in a way that proteins we just can't do with. And the bulk of my lab's work and my previous work has been trying to use sequencing and associated, sequencing technologies via in situ microscopy or standard NGS sequencing to be able to learn more about cells. And so a lot of what we do is basic molecular biology, trying to identify these relatively lowly expressed sequences and using the information gained from sequencing to learn more about these cell states. Early on in my grad work, I was doing some interesting work with multiplexing fluorophores, but nowadays we mostly just sort of do standard DNA sequencing or RNA sequencing.
So maybe we can talk a little bit about what you have found, what kind of insights you have learned so far about being able, because in this particular the latest research that you publish in which you do TCR sequencing, so if I understand correctly, you you have a slide and you are able to identify individual clones and then locate them spatially. And this gives you insight about clonal expansion, about kind of organization within a tissue, within a tumor, within a germinal center. So what are the kinds of insights that you have learned that will kind of help us understand the way that T cells function? And I mean, as I said, they are such a crossroads in immunology, and then the fact that they can see information, I love them because of that. I think that makes them so great. So what do we know? What have we learned in that sense?
Yeah, I think there's a lot of potential for learning more about human immunology with these methods. And so you brought up germinal centers. And so one question that we're interested in is how these immune cells exist in different germinal centers. Do we have different clones? Do we have clonal dominance in germinal centers on both the T cell and the B cell side? And with mouse models, you can, if you're a highly skilled lab, you can do some beautiful intravital microscopy and look at these cells and tag them. But with humans, it's really difficult to do that. And so we can start taking these static measurements by taking tissue sections and then look at the actual T cell clones that are in these different germinal centers and make statistical inferences about how these cells might be dynamically navigating in these germinal centers based off of the composition of these clones. So I think that's one example, which is we can start to identify these distinct immune niches with these spatial methods in a way that was lost before when you would dissociate these tissues.
Can you talk a little bit about how you're doing that? So how are you able to capture? I've done laser capture microdissection with caveats included. So how are you guys doing that here with the combination of, it sounds like low read is important, but also, you got to catch it from the right cell. If you blast the whole tissue, obviously it's just a pile. So how are you getting that level of resolution?
Yeah, so we take these beads. They're about 10 microns in size, which is about the size of a T cell. T cells are maybe 5 to 7. And we basically glue them onto a slide. And they're glued in this very tightly packed 3 millimeter diameter circle. Hopefully the audience is visualizing this. And then what you do is you take the tissue samples and you section them pretty thin. They're frozen tissue samples. And then you put the tissue sample onto this array of beads. And as the tissue melts, the RNA will exit the sample and bind to these single-stranded DNA oligos that are on the bead that have poly T tails to capture poly A tail of mRNA. So the reason why this is a spatial method is that each of these beads also contains a spatial bead barcode. So we can, when we do the downstream sequencing, we can say, oh, we got this RNA in this location. And we can do some additional enrichment to get the TCR and the BCR sequences.
That is so cool. No, I like it because when I think about like spatial, sometimes there's other kind of platforms that use like laser, like they point a laser to a specific spot, and then they acquire, for example, of these mass spec models, which you have basically the whole slide, and then you just use a laser to pick up a small point. But it's not like this. This is basically you're imprinting it over a series of probes, and then you're using some kind of UMI, I guess, to identify the transcripts that are binding to a specific bead beneath the tissue. I think that's really cool. But I guess probably sensitivity and things like that are probably an issue as with like single cell.
Exactly. I think it's very comparable to single cell. Mm-hmm.
And so what do you think is the next steps of technologies like this? What kind of where would you apply? What are the next steps in your lab, or you think in the field of using these very sophisticated sequencing methods to discern the position of a particular cell in the environment in which it is? What do you think are the next steps, or what are you looking into now?
Again, I think, single cell was this incredible advance for being able to answer really like two basic questions, which is what are the cell types that are in our samples of interest, and how many of each cell type are there, right? And then the advent of spatial technology has been able to answer where are these cells in tissues and how might they be interacting with other cells. And I think for us, the next big step is trying to understand the dynamics temporally of these interactions. So we started looking at some uh, contexts of aging and aging immunity in the thymus to try to see not only, we observe cell interactions at a certain time point, but if we compare a young thymus to an old thymus, what are these dramatic cell interaction changes that are happening, and what do they mean for our aging immune systems? And so I think the next big step that we're thinking about is incorporating temporal readouts as well, to better understand the importance of cell interactions in immunology.
So I wanted to go back to these beads.
Yeah, let's do it.
That's because that seems to be, I'm stuck on them, pun intended
The
here.
devil is on the details.
Biophysics, it's magic, and I like magic. Okay, no. So was that the hard part to figure out? Was the hard part like, how do I get a 10 micron bead to have poly A tails on it on a consistent way? Then when I splat it down, and then you realize that as it melts, it like sucks it out. Do you have to like add chemicals to the frozen tissue to get it to melt when it thaws? Does it just do this? Are these beads like ultra expensive and hard to get, or was this literally just putting 2 to 2 together and actually not that hard to create, just no one thought of it before, or all of the above?
Wow, that's a lot of questions.
Tell me about the beads. Like this
Yes.
seems like, key here.
Yes. So I will say when I started this, a lot of the details of this were fleshed out by Sam Rodriguez and Bob Stickles, and from what I understand, the difficult part about the beads is the density of the oligos on the surface. And so if you don't have enough DNA, you get low capture. And often when you order beads from a company, they're not that densely packed. We've done some comparisons about between these ordered beads, and now, they're making the beads in-house, sort of chemically synthesizing them. And the efficiency of capture is several times higher in the beads made in-house. So I think you do hit on an important point, which is that the quality of the beads is really important to being able to capture enough RNA. And what I worked on was on this TCR sort of low expression transcript side of it. It's even more important that you have these very densely packed beads, because if you miss out on capturing them in that initial step, you'll never be able to recover them in downstream sequencing, even if you amplify. And so for us, trying to figure out ways to a little bit of optimization on the tissue side as well. You want sort of fresher samples. You don't want samples that have been frozen for a decade. But really, it was just figuring out, now that we have beads that are capturing the TCRs, how we can enrich for them and different methods for amplifying them in a library.
I want to kind of switch gears a little bit and talk about your career and the way that your research, I guess, because you're maybe not so much you just mentioned you're originally more a biophysicist than an immunologist, and so I guess that a lot of the projects you're carrying on you're collaborating with other researchers, and of course your particular geographic position is excellent for that kind of work. But so I mentioned before when we started talking that you're one of those people that went right after grad school to have your own research group. And I think that's something that we don't see a lot nowadays, although I do see that several institutes are offering this position that is a bit more visible. So maybe we can share with us a little bit your experience of moving from being a PhD student directly to having your own group, and what do you think has been the main challenges so far? And also, how do you think you have used your knowledge here? You're mostly trying to collaborate, or how have you found your way collaborating with other researchers in order to apply this kind of platforms that you're developing?
Yeah, so I guess there's two sort of origin stories of my experience in this early independence path. And the first is that when I graduated or was about to graduate, I was actually so excited about doing a postdoc. I think a postdoc is a time where you're highly skilled, you're able to run projects, and I was so thrilled at the aspect of doing that. And then I was at a conference where I met Ron Germain, and we were talking about diversity in science, and he brought up this paper that I thought was so interesting. It was this paper by the NIH in, I think, 2007. And they basically surveyed women from the transition between postdoc to faculty positions, and men as well at the NIH. And there were two sets of questions. The first was questions that were like, how are your skills at designing experiments, doing controls and analyzing your data, rate from 1 to 5, so on. And the other set was, how confident are you in getting a tenure track job and succeeding to get tenure? And it turns out that when you control for women and men responding the same way about their skills, women basically say they have much lower confidence in their careers. And when Ron told this to me, I was fascinated because, I was so excited about doing a postdoc. That's all I was thinking about. And that opened my mind to thinking more like, I have a vision for what I want my lab to be like. I know what I want to do. I'm so excited about these tools that I'm building. I'm going to pursue this. And it encouraged a sense of confidence in me that I really want to share with. I think everybody needs a little bit of confidence, and especially folks in science, when we're not always the best at selling ourselves. And so that, I think, was how I got excited about the idea of starting my own lab. And the other part of it was, my advisor was an early fellow as well at the Broad, and seeing it happen made it seem less scary. Being his first graduate student and starting things up made it more tangible. And I think a lot of the unknown can be quite daunting to people. So when I actually started, it's been like 2 months. I think the most challenging part for me has been knowing when to pause and take a break. Do you guys know who Haruki Murakami is? He's this author. He writes a lot of fiction. Yeah, he's written like Kafka on the Shore and so on. And he has this book about running, and he talks about how basically when you finish every day, it's important to finish at a point where you're still excited for the next day. And I like that a lot as it applies to research, because in the first 2 months, I've had this alarm on my phone where I need to go home at 8 p.m. Even if I want to stay later, I just have to get up and leave, close my laptop. And then, sometimes I'll even say out loud to myself, like, I'm happy with the work that I've done today. I can go home and be excited for tomorrow. So it's not the challenge that I thought I would have. Everyone said, oh, hiring will be hard because you're so young. And I've been very fortunate to recruit some really great people. And so the surprising challenge for me, I think, is just in pacing myself. It's a marathon, not a sprint, I guess, as they say. And then on the collaboration side, I think working with people has been one of the joys of science for me. It's, I think, the source of where really creativity comes into play, when you can have two seemingly distinct areas of expertise come together to find a really nice overlap. And you're right that Boston is great for this. It's so dense in scientists. I go on the subway and I hear people talking about chemokines, and I'm like, yes. And it's a great place for that. And I've been very fortunate to work with some really nice people. And honestly, that's the most important thing, I think, when I try to find people to work with. Lots of people have nice samples, have really great questions, but finding nice people to work with is the thing that brings me joy in science.
So can you talk a little bit about this special program you have, this early investigator independence thing? I think many people have no idea what those words mean put together at all.
No, I forget, because my advisor Faye was a fellow, and so it's been sort of almost like ingrained in my training that was part of my experience working with a fellow. So in the US, there are maybe around like 30 or so positions a year. And the idea is that they take folks out of graduate school and they provide them with a startup fund to be able to hire a couple of people to fund their own research. And there's usually a time constraint of like 3 to 5 years or so. for some of these positions, they are on some track to getting a professor position, but I would say the vast majority of them are not on that track. It's more just to support young scientists and their crazy ideas. And there's been some analyses on the outcomes of these scientists compared to folks who do the traditional path. And there isn't any sort of worse outcome for these early independence fellows. They're cited quite similarly in their career. It's just a different path, and I wouldn't say it's better or worse. I've talked to many people about it and having experienced it. It's a lot of work. It is a lot of fun, but it's definitely for people who are really focused on trying to build something straight from graduate school.
It sounds like a really great opportunity. If you, as you said, if you already have the idea in your mind crystallized enough, you just need that support, right? That funding and that maybe some training, maybe some administrative help. I don't know. That can be also quite daunting. I think probably more daunting than the science itself.
Oh yeah, absolutely. Like the science, I know that we can do it, but finding the right support, I think, is so essential. And logistically, it's also it's a very different path, and in a lot of ways, scientific community can be very resistant to that path, right? And so when you're evaluating people who haven't done a traditional postdoc, I think it can be a little bit harder down the line. But for me, it's been a good time. Folks at the Ragon are really incredible, very supportive, and that's what made me feel very confident in starting this.
All right. So next up, I guess for you is finishing getting your stuff together, get it and no, I guess you're 2 months in, fairly still a lot to do. And continue looking into interactions of T cells and immune cells in tissues. In you mentioned, so you just mentioned about your some work on thymus and aging. Um, what do you think? So when you say old thymus, what are we talking? Are we talking like 30 old, 60 old? Do we even have a thymus when we're 60 anymore? In the human situation, how does it compare with mice or with other better known models?
Yeah, so in humans, our peak thymus is around like 16, and then it has a half-life of just about the same, like 16 or 17 years. So at around age 30, your thymus is about half what it was, and it's replaced with fat. And yeah, it's very sad. I see your sad faces. That's me on my birthday every year. Um, and so I think we're interested in this because of what the implications are for immunology as we get older, right? And our immune systems. And there was a great paper recently from a collaborator of ours from David Scadden's lab, the New England Journal of Medicine, where they find that removing the thymus can increase your all-cause mortality and risk of cancer in people. And I, from what I've seen with collaborators up until this point, often thymus would just be discarded as tissue that was in the way of the heart, right? I know, and the scary face is real.
Horrified face.
This is
No!
the real Halloween fear. But so yeah, I think we're really interested in one, how this process is happening, and two, can we figure out ways to repair the thymus to enable it to generate T cells with age? And there are actually some really interesting contexts where this happens. So during pregnancy, the thymus will actually shrink. And then after giving birth, the thymus will grow back. And people think this is in part because it's important for mothers to tolerate paternal antigens in the fetus. But this regeneration process is really interesting to me. And trying to look at these contexts of regeneration as ways to potentially think about regenerating thymus in the elderly to strengthen vaccine response, I think could be really cool. But more generally, I think there may be like 3 sources of aging. The first is, are there changes to the original progenitors that are leading to an aged phenotype? The second is this organ context. Do we see changes in the thymus or in the bone marrow that make it difficult to mature these cells? And the third is in the periphery, where maybe repeated exposure to things like cytomegalovirus can constrain our immune systems with age. And for now, we're interested in the tissue context, but hopefully we can expand more broadly in the future.
I wonder if it's on purpose that we this degradation of the thymus, whether it's because we never made it that old, by in evolution, that didn't really matter.
No, we did. So people who made it past like early adulthood often lived into their 60s, even
Well, but they didn't have
thousands of years
children.
ago. No, they did. They had children and then made it to their 60s.
Yeah, but not after like their third. Well, maybe at their 30s would make sense.
Oh yeah, I think this is super interesting, and I will say there are some like interesting exceptions. There are some species of sharks that have really like prolonged thymus size with age. And there's some papers about like ectopic thymus, so basically alternative sites to generate T cells. But I think this is a super interesting area and has been the subject of many late night conversations.
That I'm intrigued. Maybe you can just grow a new thymus. Like, oh, you're having your responses to vaccines are getting pretty low lately. We're just gonna insert your new thymus in your thigh or something. Like they grow the organs.
Yeah, there are companies that are trying to do that now. So maybe we'll see in the next few decades. Yeah.
But see, I always say after 30, it all comes downhill from now. People don't believe me, but now I have a very specific example. So just beware. It comes to all the time out.
With age comes wisdom. So looking forward to that.
There we go. Something positive here. Come on. I'm sitting here being like, some of us are in our fourth decade.
You're very wise, Jason. Very wise.
Right. Exactly. Don't you forget that, Brenda. I'm the wise one.
Yeah, I would say luckily our peripheral diversity does a pretty good job for us actually up until like 50, 60-ish. So you have a couple good decades left for you.
Yeah. That's great.
You know what? Let's just end this here on that note.
Put me out to pasture already.
We'll take it to a farm upstate in a couple of decades. Don't worry. So why don't we go to the last part of our conversation? Jason, do you want to do the honors?
Sure, I can still remember it. So, we always try to do a little fun question here. So for you, if you weren't doing all this cool biophysics magic, what would you be doing instead?
Well, I really like space. I think trying to figure out ways to terraform other planets would be really exciting. And I think actually, well, this interest in space has come from reading a lot of science fiction. I'm really inspired by like Isaac Asimov's career, where he was a biochemist and a professor, but also sort of pivoted to really focusing on a science fiction writing. And so maybe science fiction writer, maybe science fiction actual scientist, I guess still a scientist, but a different kind of science, which I think would be very exciting.
All right, I got to ask, have you played the board game Terraforming Mars?
I have. It's so much fun. Highly recommend. Not sponsored.
Yeah. It's a very fun. And I guess you've read The Martian and all those.
Oh, yeah. Yeah.
I just want to say that I'm also a big fan of Isaac Asimov, and also very much appreciate his career trajectory. And so I think 100%.
Nice.
I highly recommend a series that most people haven't heard of called the first one's called The Unincorporated Man. So the solar system's colonized. Someone from our age cryopreserves himself with a deadly disease and is a billionaire, multi-billionaire, and is only sent into is preserved until there's a cure for it. So he wakes up like 100 years later into this new society. And it's ultra capitalistic in that every person has shares of themselves. Your parents get a partial ownership and you have to auction off your shares to fundraise. Want to go to college? Shares of the institution you go to, so on and so forth. Well, he is unincorporated because he was born before that and all the laws are still on the books. And he also has a bunch of old Earth artifacts like with him, like baseballs worth tons of money. So he's ultra rich in the future and unincorporated. Enter science fiction. It's a hard sci-fi, right? So it's like, no faster than light travel, but there's mining of the asteroid belts and stuff. And what do you do with a dude who's not a corporation in a world where literally every person's a corporation in science fiction land?
Very interesting. American future dystopia.
And
that sounds really cool.
It's very interesting. Unincorporated Man. It has a little bit of feel like The Expanse to it.
Oh, I love The Expanse. Yes, we should do a science fiction podcast.
Sounds fun. Then we should invite, as we mentioned before, the host of our sister podcast, the STEMCELL Podcast.
Arun.
Also,
yeah, space
Arun
science.
is very much into space. So
like sends experiments into space into it. and spoke to
Yeah.
Cameron Harris about it.
Do stem cells grow in space? I don't know. He'll find out.
As someone who studies like spatial biology, I'm always missing out on the fun space.
Yeah, spatial, not that space. I
Not
mean,
that
like
space.
the organization, a spatial organization. Yeah, I know. It's like being a doctor, but not that kind of doctor. The PhD kind of doctors, like
You can just do both. Why por qué no los dos?
like you, of course.
See, well, so you can do this and do space science at some point. It can happen.
All right. On that note, was such a pleasure talking to you, Sophia. Thank you so much for being today on the podcast. And good luck with continuing your research and building up your team. And we'll see what comes up next.
Awesome. Thank you so much.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on Twitter at @immunopodcast or via email at info@immunologypodcast.com with feedback or to suggest guests. See you next time.